introduce our speaker, Dr. Dama. I'm sure many of you already know him, but for those who are not, he is one of the rising stars in the field of Alzheimer's disease and their application with AI. He's an assistant professor of gerontology and geriatric medicine at Wake Forest School of Medicine and jointly affiliated with the Center of AI Research. He obtained his PhD in medical and biomedical imaging, competing from University College London, and did his postdoctoral training as an Alzheimer's Society Research Fellow at Simon Fraser University, specializing in artificial intelligence applications specifically for Alzheimer's disease. His research evolves around developing explainable machine learning frameworks for multimodal medical data integration to advance early detection, precision diagnosis, and disease subtyping in Alzheimer's disease and related dementias. He was recently awarded for two major grants, one from NIH with a form of R01 and the other one from Alzheimer Association. In this talk, he will represent his work on leveraging multimodal neuroimaging to improve early detection of AD, coupled with explainable graph neural networks that capture heterogeneous cortical neurodegenerative patterns in a clinically explainable manner. Please join me in welcoming him. Dr. Da, the stage is yours.
Thank you so much, Abraham, for the very nice introduction, and appreciate for all the openings. And so, hello everyone. I will start my talk today, and as Abraham mentioned, the title is called AI-driven integration of neuroimaging genomics for Alzheimer's disease and brain aging. So I just, before I start the main talks, give some background about why Alzheimer's disease. So as most of you probably know that Alzheimer's disease or ADs is a leading cause of the dementias, especially in the aging populations, with the affected populations in the elderly populations will be over 55 million globally. And as you can see on the right, there is a multiple biomarkers that can be detected at different stage of the disease. However, the early detection of the dementia still remains challenging, mainly because of the complexity of the disease onset, especially its multifactorial risk biomarker profiles. And then why do we use neuroimaging and why genomics? So recent years, we have renewed the definition of Alzheimer's disease as ATN framework, mainly following the disease progressions from amyloid, tau, neurodegeneration, and finally cognitive declines. And because all these biomarkers can be detected decades earlier during the progression before the clinical symptoms, and so neuroimaging data has been the hallmarkers that can be used to detect different AD-related biomarkers, for example, the amyloid and tau depositions and the neurodegenerative patterns. And on the other hand, not only the biomarkers are very precious, AD is also considered as highly heritable. The twin studies recently showed that about 60 to 70% of the AD risk are heritable. And so basically we can build a polygenic risk scores for that. The reason is that although with a higher heritability, each individual loci has a very small effect size when trying to predict the diseases. That's why we need to identify a global risk factors to identify novel treatment targets as well as the patient stratifications. And that's why that's the background of these talks. And move on to the first part is about the neuroimaging. And what we want to present here is using multimodal neuroimaging fusion approach to improve the dementia detections. As we mentioned earlier in the graph, which I showed again here, that for example, PET imaging and MRI imaging are two hallmarks which actually representing different disease stage of the imagings of the AD progressions. So while earlier studies have only been using one modalities, some example studies, for example, I showed here in our previous research showing that combining both imaging modalities are actually improving the disease risk score. So how we can evaluate disease is also important. Most of the time, people only look at the sort of a classification, or we call it identifying the AD versus the cognitive normal, or NC. However, if we want to do the early predictions, we would like to stratify the patients in a more maneuver or more stratification levels. For example, considering their current diagnosis and future diagnosis, we can stratify them further into the progressors, as shown on the bottom, and non-progressors as shown on the top two. And then there will be 7 stratification groups. And as shown here in our previous studies, those stratification groups should have lower to higher disease risk as the disease progress. So on the x-axis, it's the disease risk from 0 to 1. And you can see using MRIs, we can see the stable NC has the lowest disease risk, and then further down to the stable DATs, which have the highest risk. And our earlier studies shows that amyloid PET can have the same detective values. But if you look at the stratification or look at the separations among different groups, you can see that actually there are the strongest separations that happened in the earlier stage. However, there's some distinction. There are some discrepancies or some part or confusing part when looking at the progressive NC and SMCI, and when we are combining both together, so potentially the disease risk stratification will be better, and this can be coped with the different explainability methods, such as guided backpropagation, as I showed bottom, indicating that different image biomarkers indeed looking at different parts of the brains. So with that in mind, when we are applying those multimodal neuroimaging data or models into the clinical applications, then several challenges arises. First is that how can we know that we are actually effectively fusing these cross-modality informations, especially they are representing different complementary informations at different stages, and what's the metrics that we can evaluate that? And secondly, a lot of time in the real world data, not all, or if only a few of the subjects having the both modalities available, because depending on the disease stage, the doctors might decided to acquire only one modalities, and how we can cope with that, and how we can still use multimodal informations in this scenario in real world cases. So that's what we developed here, is that first we developed some a novel loss function called cross-modality self-attention and deep canonical cross correlation, where we're trying to maximize the intermodality correlations in canonical feature space. And the second is that to directly dealing with the availability of a partial image modalities, we developed an intermediate layer called modality-aware mask-guided attention, or MEMGA, where during the trainings, we randomly mask out one of the modalities. So in this case, the attention mechanism, we're trying to learn the joint probability, it's like the mask autoencoder, where we're trying to increase the difficulty of the problem, however, giving the true answers during the training, so that the models can learn both modalities, while during the inference, it can, when it only gives one modalities. And the result shows here is that, so you can see when we are combining both modalities together, the predictive power is increasing. However, if we have a better multimodal fusion strategies, the prediction power further increases. And when we're combining that together, we can actually having a better predictive powers when using both strategies. So the conclusion is that there's a way to design a flexible multimodal neural fusion strategy that can improve upon the state of the art, especially in the real-world clinical applications. Not only is the accuracy matters, but also the feasibility matters. So with the limited modalities, we showed that we can leverage a attention-based cross-modality information fusion approach to adapt the model, the previous retrained deep learning models into a real-world clinical scenarios. And second part is about, again, another part of the clinical application challenge is not only the data limitation, but also the clinical explainability. And I'll put an example of our previous studies to show some conventional approach to do the explainable AI studies. So feature engineering approach has been proposed for decades, where you can, for example, in our studies, we can do the parcellation of the brain structures to extract the handcrafted features, which we can then harmonize the features and then derive the feature scores. And then the feature importance can either be embedded in the data, in the models, or can be do post-hocly. The issue for that is that because the extracted feature actually causes a loss of the spatial information, and also it doesn't retain the anatomical or pathological features across the entire brains, so it will release the suboptimal predictive powers. On the other hand, the feature agnostic approach, such as the convolution neural network-based deep learning approach, has shown the better predictive powers. However, because of the black box features, there's a little studies or few ways that we can harmonize the feature during the trainings, as compared to the feature engineering approach, and also because of that, the explainability can only happen post hoc, limiting its powers. So in order to address those the limitations or the pros and cons from both approach, where we are designing a explainable cortical graph neural network features and deep learning predictive models. In this sense, we are doing a search of the pre-processing steps, where we model the brain corticals as a surface which can be considered as a graph, and then we can use a graph neural network which is intrinsically explainable. And in these settings, the edge of the graph will be the surface mesh topology representing the entire brain anatomy, and then the node will be represented by the vertices on top of the surface mesh, and at each vertex on top of the edge graph. And the node features can then be defined as either the atrophy morphologies, such as the thickness, surface area, or curvatures, as defined here, or we can add in even more modalities if we can co-register the images together. And the strength of this approach is that, as I mentioned, the graph neural network is intrinsically explainable, and that's why the clinical relevance is guaranteed. And also, more importantly, it solved the problem of that not only we can now handle the non-structured spaces like 2D manifold, whereas the traditional convolutional neural network cannot handle, but also through the atlas-based registrations, we can now effectively harmonize the features and removing the confounding factors such as the site and scanner features, as well as the demographic features as sex and age, for example. And however, there are still challenges imposed in this approach, where it's super computational intensive in terms of the pre-processing pipelines, and also such predefined graph has a relatively shallow feature, which as represented here, we only have 3 node features, but having a very condensed the nodes representing a large graph. So how to handle the massive graph features and embedding the graph in an effective way is a challenge. So here on the right side, I'm showing the stepwise approach that indicating that each steps of the processing pipelines, and I'll talk about especially the design of the graph embedding features and as well as the explainability part. So in total, we tested 4 different graph neural network features on top of that, and baseline models, which is MLP models. And as you can see that the graph convolutional network shows at the moment it shows the least performance, given that although it's scalable, efficient through the convolution operation. However, the over smoothing causing the problems. And progressing to the best models, are graph transformers and also the graph attention networks. In both cases, we are trying to utilizing or injecting the attention mechanism in the transformer stage, where the graph transformers are using global attention, meaning it can capture the long-range global structure dependencies. And comparatively, the graph attention network is is able to capture the local attention where it captures the nearest nodes or neighbor nodes dependencies. And because of the highly dependent feature, highly dependent characteristic of the node feature in the graph, actually the graph attention network shows the more effectiveness in terms of getting the local dependency features. And now that we have the predictive powers, we can now do the explainability approach, and to look at what is the important feature that representing the better predict powers. And what is shown here is interesting, that not only can the explainability approach dedicated to show that the areas like the temporal cortex regions are highlighted as the for these individuals as an important features that showing it's related to AD related features, but also it can also compare different types. For example, it shows that the thickness features are actually more representative or more important or more crucial compared to the curvature and the cortical areas features when doing the predictions, which actually makes sense because that's usually the features that most used. But now we have a visualizable way to prove that is indeed the correct selection of the features. So in conclusion, so here in this project, we showed the effectiveness of harmonizations for confounding factors when using graph neural network features. And also by comparing different graph neural network architectures, we showed that the local attention showed very effective powers to aggregate morphological features compared to global attention mechanisms and other different graph aggregation or message passing frameworks. And this is just the starting of this kind of the approach where, for example, we can start to think about hierarchical graph pruning to effectively further perform the node feature embeddings more efficiently. And we can, as I mentioned, we can add in multimodal neuroimaging and cortical neural features onto this to make it a multimodal models to have a better predictions. And also we can also using the graph autoencoder and other related approach to do the clustering approach on top of the classification approach to look at the subtype of dementias, which will be useful for precision medicines. And moving back to the other side of the story about the AD progression or AD etiology, which is the genomic part. So as just a recap, as we mentioned in the first slides, is the genetic mutations or genomic risk factors account for a large proportion of the AD risk factors. Why do we study genomics, however? The reason is that if we identify the important genetic variations or mutations, we can then study the function causal pathways, which can lead to potential drug to targeting different pathways, for example, inflammation or cardiometabolic pathways. And however, conventional way to handle the high dimensional data for the genomic data is to generate the polygenic risk scores is usually using the genome-wide association study, in which case each of the genomic loci are studied separately or independently to look at their associations to the AD risk. But in that case, this approach is subject to linkage disequilibriums and also is unable to capture the gene-to-gene interactions, or it's called epigenetics. And that's why we are trying to build a deep learning-based polygenic risk score models. The advantage is that it can capture the complex gene-gene interactions and can handle the high-dimensional sequence data with the potentially long-range epistatic gene interactions. And then it can, on the right, I'm showing the pipelines of these proposed pipelines. The main challenge for handling the deep learning, handling the genomic data using deep learning is the high dimensionality in the one-dimensional vector. So we are proposing a cascaded approach where we are doing a sliding windows feature selection first, and then apply a deep polygenic risk score predictions, and then followed by a explainability approach. So I'm showing the hyperparameters that is important in the first step to fine-tune what's the best sliding windows that are used to find the best parameters to predict models. We use balance accuracies. We select the best balance accuracies, which is balance between the sensitivity and specificity, and using the validation set. And when applying it to the independent holdout testing set, the result shows that the deep learning-based polygenetic scores showed significant improved power compared to the polygenetic scores. Noted that although the accuracy seems like lower compared to the imaging data, but that's the nature of the genomic data, which actually is confounded by a lot of lifestyle changes. So this is very reasonable as within the state-of-the-art range of the predict powers that the genomic data can handle for the dementia risk. And further on, not only do we want to predictions, the most important thing is to trying to, as we mentioned earlier, trying to identify novel genomic loci or novel target for potential drug treatment designs. And so here we are using explainability approach to actually first is using the model agnostic approach, where we are doing the feature occlusion by occlusion each features sequentially and identify the features that are most affected by the occlusion. And the second is using a deep learning specific approach called backpropagations. And when we are listing tables to show this sort of a pseudo Manhattan plot or pseudo Miami plot, we can see that there are some overlapping loci that can be identified by both approaches, but also there are some independent loci detected. That interest finding indicating that potentially the selection of the approach we are used for clinical explainability matters when we are trying to identify different pathways. So here is a summarization of the two tables where we can identify a cluster of genes or gene loci which has been well established in the previous research. For example, the ApoE is the strongest AD risk genes as we know so far, and the ApoC1 is in linkage disequilibrium with the ApoE, and TOM40 and nectin are both related to different pathways. One is to energy and or mitochondrial dysfunctions. The other is through the immune system or blood-brain barrier breakdowns. On top of those known genes, we also identify some novel genes that was either secondary or a potential candidate for target gene-related drug targets. And so, for example, DOX6, TSPAN, and ZNF766, PDE4C, BCAM, TLE2, and C3 are a couple of the candidates we listed, which we selected only the top 20 of them. And if we grouping or clustering those genomic loci, we can see that all the known loci are around the ApoE locus regions. And on top of that, the additional genes can be reflected into, or can reflect or be categorized into different signal pathways, highlighting the synaptic, vascular, immune, and the signal pathways, immune signal pathways, offering a good candidate therapeutic target beyond the known ApoE effect. And so in conclusion, so here we are showing for the genomic, in the genomic risk factor settings, the deep learning has demonstrated significant improvement powers compared to the conventional GWAS-based PRS. On top of that, we also elaborated or showcased some potential ways to using multiple explainable AI approach to reveal not only established AD risk loci, which is you can consider as a validation studies or validation cohort, but also we can identify normal candidate loci as a potential treatment target. So this is not this is only starting of this kind of line of work, and so we are only exploring one type of the deep learning architectures, potential novel architecture. For example, temporal convolution networks or the transformer-based genomic language models would be very suitable to handle the sequential data, such as the genomic data. And then care need to be taken when we are trying to tokenize the genomic feature into language models, because the genotype array data is very different from the whole genome sequence data, where the raw sequence is shown. So a lot of researches need to be done in these areas. And also, the identification of the genes will now open the door for us to do further pathway analysis studies. And also, we can look at for each individual genes, we can look at their clinical and biological relevance, for example, by looking at longitudinal survival or functional annotation association studies, and which can potentially lead into the design of the future clinical trials. So in summary, we show the genomic-only risk factors that can be used in AD predictions. However, combine these two together, if most of the existing studies are similar to what I've shown previously, looking at the neuroimage data independently or the genomic data separately. And however, if you can see that the genomic risks are actually, as you can see on the figures on the right, there's a dynamic influence of genomic risk to different biomarkers or different endophenotypes at different stage of the disease. So why not combining them together? Potentially integrating the analysis of the neuroimaging and genomic data can give us a comprehensive view of the disease etiology, not only cross-sectionally, but also dynamically across the disease progression or for each individual subjects or potential at-risk populations. And also the explainable AI approach can now analyze the joint features and reveal the relative importance of the contributions between the risk and the biomarkers landscape. So that's exactly what we attempted in the next step, is that here on the left, what we are showing is first rows is the multimodal neuroimage-based approach to detect the dementia risk. And the second rows is the genomic-based risk factors. And then in the third rows, we are using a multi-level, multimodal feature fusion or embedding approach to get the neuroimaging genomic disease risk factors. And on the right, we are showing the comparisons of these three different models. And on the top, you can see that progressive NCs is showing the lowest risk. However, in the bottoms, you can see that progressive NC is showing a progressive NC should have a higher risk. However, it's not showing there because of the disease stage and the limitation of the data modalities. While combining them together, we can actually potentially get a better risk prediction for these specific stratified disease risk groups. So in that settings, we can see that when predicting disease risk, combining the neuroimaging and genomics give us a better sort of tools that we can use. And on top of that, again, back to the clinical requirement of clinical applications, not only are we interested in terms of the future risk, but a lot of times we are interested about the time to potential future disease onset. In the setting of the Alzheimer's disease, it's about how many years when at the time of the diagnosis would we expect the patient to convert to dementias, and that has a lot of implications to the future management plannings and potentially lifestyle interventions. So here I'm showing the design of a survival analysis approach where we can actually inject the deep learning-based approach to derive a survival analysis where the person during the studies, where the person who converted from cognitive normal to AD during studies are the subjects of interest in our study. But however, the people who converted before and after are considered so-called census data can also be used in the survival frameworks. And so using these frameworks, we can develop called hazard ratios, where we can look at both the genomic risk factors across populations and also the neuroimage-based hazard ratios to derive a disease risk as a function of time. And then using a preset threshold, we can then derive a time to dementia onset. And compared to the sort of a populational level hazard ratio in the genomic data or the traditional statistic approach, in these predictive frameworks, we can not only get a population-wise survival curve, we can also derive the individual dementia risk survival curve. And using that settings, we can actually having an individualized time to dementia onset event predictions. And by comparing the true onset time, because we have a longitudinal follow-up, by comparing the true and the predict power, we can now start to train and evaluate our deep survival models. So again, coming back to the previous framework where we are comparing the neuroimaging genomics and neuroimaging genomics integrated models. But this time, instead of only predicting the disease risk, we can start to detect the risk as a function of time. So there are two. Here I'm showing the two evaluation metrics to look at the survival prediction accuracy or performance. The first is the concordance index, where the bigger is better. Here it's showing that the fused models, the MRI and SNP's fused models shows the better concordance index. And similarly, the integrated Braille scores is showing the index in which the smaller values indicating a better predictive performance. Again, the fused models give a better performance. And coming back to the clinical implications, in clinic where we are interested is actually whether the predicted onset time is actually before or after the actual time, which actually is different. So in these settings, when we compared we further stratify the patient into currently cognitive normal versus currently mild cognitive impairment group. So if we look at using the genomic data, basically SNP-based studies, the people who are currently cognitive normal shows a good prediction indicated by the very close to the zero differences between the predicted and the actual disease onset time. While for the mild cognitive impaired patients, the predictive is actually longer time, meaning it's false negatives, which is crucial or more less favorable compared to the false positives. Well, compared to when we are looking at the MRI-based models, interestingly, the currently diagnosed as a normal cognitive patient, they have a very bad performance in terms of predicting their future dementia onset, which makes sense because their biomarker or image-based biomarker hasn't been reviewed. However, when at the later stage, during MCI stage, their predictive power is actually better compared to the genomic data, and that indicating that different risk and biomarker factors are actually affecting disease at different stages. So potentially combining them can give us a better results. So again, since we are focusing on the clinical relevance here, so now having a combined models, we can start to build a relative feature importance maps, not only for each individual biomarkers or risk, but we can have a composite risk factors. So here, the neuroimaging based features are shown in red, the genomic feature are shown in green, and also we added a clinical and social demographic factors in the models to mimic the real world applications. And you can see that as can be expected, the cognitive features having a highest say to the future disease risk of dementias, followed by the neuroimage data. But then gradually the genomic data actually consist more and more importance in the later part of the feature importance maps. So in conclusions, here we are showing that the neuroimage biomarker and genomic risk can be combined together and demonstrated not only the joint predictive power, but also dynamic progressions of the disease stage at the different progression spectrums, and integrating them together can improve the prediction of dementia risk as well as the time of onset. And the future work of this is also, there's a huge opportunities where we can start to build the stage-aware diffusion method, and then we can also using similar to multimodal neuroimaging data as the study was showed, we can use attention-based approach to have a better fusion strategies. And also, as we just allude here, that additional biomarkers like novel fluid biomarkers, like plasma biomarkers, or additional risk factors like lifestyles related risk factors, such as cardiometabolic risk factors, can be added into this framework to have a better clinical outcomes. And since we are talking about the longitudinal predictions, another aspect we would like to evaluate in the real clinical settings is that if patients are followed by several years or several visits, can we utilizing this sort of a longitudinal input data to further improve the predictions? And if that's the case, then potentially we can design the screening test or designing the early prediction test in a more efficient way, giving the purposes. So here, what we are showing is that we using we concatenated 3 follow-up scans from the initial visit time point so first we embedding the features for each time point separately, and in the second stage we are using a simple convolutional neural network to learn this time-dependent feature. The reason using CNN is because 3 time points CNN is sufficient. We suppose it's sufficient to cover the time dependency curves, and then make the predictions. And we are happy to see that after using these strategies, when we concatenate the 3 time point data, the MRI only based models showed improvements. Not only that, when we combine the MRI data with the non-MRI data, it also showed improvement. And when we are doing the time to onset survival analysis predictions, the improvement seems like even higher when predicting the MCI to AD converters, but also the CN to AD conversions. So that concludes the sort of AD disease based studies. Another thing we want to jump out of the disease risk prediction from the dementia point of view is that all the early predictions nowadays, we would like it to be the preventive care in the sense that we would like to see the patients, or we would like to see the population or subject at a earlier stage where there's no onset of no signs of a disease, but maybe some at-risk signs. So we want to see whether we can looking at the earlier stage by looking at the healthy aging. So in these settings, what we are trying to do is to building a biological clock or aging clock. So some definitions here. Chronic aging is defined as a time since birth. Biological aging is actually what we would like to see is a function or status of the entire sort of body's molecular level and phenotype level signatures. And we want to use a deep learning model to learn from a chronic age, but then predict biological age. So the differences between the predicted age and the actual age, or we can consider as an accelerated biological aging, and that's deviating from healthy aging trajectories. That could reflect the higher risk of potential comorbidities like cardiometabolic disease, cancer, and neurodegenerations. So in that line of studies, first we tried to use the DNA methylations to build a epigenomic clock. The reason is that DNA methylations represent a lot of the lifestyle or environmental effects on the genomic data, which highly representing the aging process. So we are using the temporal convolutional network, which combines the temporal features with the dilational blocks to build a aging predictive regression models. And you can see that the predictions, the correlations between the predicted age and the actual age showed relatively good correlation. And when we are embedding when we projected the embedded features of the age predictions, and then we color-coded that with a different age group, we can clearly see that different groups are being clustered into different regions. Noted that although I said there's a good correlation, you can potentially see there might be a imperfect correlation happening there, meaning that there's a negative correlation compared to the sort of when comparing the predictive and actual brain age. And I will mention that. So this is a general phenomenon when we are predicting both the epigenetic aging and the neuroimaging aging. So I will give you another example using neuroimage-based brain aging. So in our previous studies, we are using the T1 MRI-based the data to predict the chronic age, and then we are only using the cognitive healthy subject, and then we can apply that to potential at-risk populations and see their disease risk factors and see whether there is a associations between the accelerated brain aging with all these factors. And we indeed identified some of the some of the risk factors that are potentially risk of accelerated aging. So here is also again a plot of the chronic age predictions for using the brain image studies. And here I showed the overlap of the perfect correlation map with the actual correlation map. You can see the difference. So this is where we are trying to see whether we can make further improve these brain aging studies or biological aging studies, that we want to explicitly remove or penalize the negative correlations that's happened during the trainings. And it's a global phenomenon that has been observed in a lot of regression studies, especially aging studies. And at the moment, the well-recognized approach is to do a post hoc correction, meaning that people will doing a linear fit of the predict data and then do the corrections on the validation or test set. The drawback of that is that it's subject to the bias between different training cohort and the discovery cohort or the validation cohort, and it's not generalizable in terms of when the population has changed. And so here we are designing a loss functions, which is we call the negative correlation loss, and combine that with the mean average mean absolute error, which is the default loss functions used in most of the age prediction tasks. As you can see on the left, so when we are adding more and more of the negative correlation loss in the trainings, the MAE loss start to increase. However, before it's jumping up, we are able to find the optimal values that can minimize the negative correlation to almost zeros. And showing on as a figure on the bottom, if you can see the negative bias as opposed to the chronic age, you can see gradually we can start to remove these negative bias during the training, representing the effectiveness. And another thing we also tried in the ablation study is that because of the normal distribution of our populations, there's an uneven distribution of the agent group. And if we do the age stratified rebatching, and then you can show here on the left is showing the without batching, and on the right showing the without batching. So the correlation is actually better whenever we have the age stratified batching. And similarly, the ME is actually lower in this case. So adding both methods together, we are able to to solve the decades-long problem of the negative correlations in the regression problem, especially in the aging predictions. And the future work of this line of studies that we can start to build an explainable AI approach to understand the brain age patterns and to stratify different aging population or different aging trajectories. And also, we only consider the cross-sectional data. With available of longitudinal data, we can start also to inject the longitudinal components and see whether we can, using the inter-subject longitudinal age discrepancies, to further improve the models. So in summaries, here what I'm showing in different projects, but overall, I want to have the overall take-home message. I listed that into three parts. The first is that multimodal data fusion is important. The examples we showed here is using MRI and PET and neuroimage and genomics. And then it can improve the applications for both time of onset predictions and also biological aging. But then novelties need to be addressed to handle cases where there is a missing modality informations. And second, as you can see throughout the project, explainability is one of the most crucial parts when we are considering the clinical translations. Either it's using neuroimaging data, raw neuroimage data, graph data, or genomic data. And then when we are having the multimodal time varying dynamic feature importance, it can be embedded when we are aggregating different modalities together. And thirdly, for the clinical translatability, several levels of the approach or the cares need to be taken. The first is that to ensure the generalizability, feature harmonization is important wherever it's possible. And also any biases, even seems small, will be important to remove when we are looking at individual like age, like biological age predictions. And the second is that when we are dealing with the longitudinal data, it will be beneficial to incorporate those informations and then having a more clinical relevant outcomes, for example, the time to onset rather than like a direct risk predictions. And third, stratification is important to have to understand the population at different risk stage. And finally, in the real clinical world settings compared to the cohort data when we're recruiting patients, there's a lot of missing data. In these settings, the entire modality will be missing, and how to handle that is important to make the translation between the AI development to the real clinical settings. And with that, I would like to thank all my lab mates and also collaborators, mentors, and my funding bodies. And thank you everyone for listening. And I'm happy to take any questions.
Wonderful. Thank you, Dr. Ma, for your wonderful presentation. It looks like we have a hand raised by Dr. Gherkin. Would you like to unmute and ask your question?
This is a fantastic presentation. It's really exciting. So congratulations.
Thank you.
My question is about the biological age versus chronological age. That is really interesting. You might have mentioned this, but do you see this gap between the biological and chronological age much larger for Alzheimer patients versus non-Alzheimer patients? Also, do you see that gap getting larger when you go from non-Alzheimer patients to MCI, for instance, and then Alzheimer patients? Do you see these kind of trends?
Totally, yes. That's a great question. Thank you, Martin, for picking that out. We actually that's exactly what we are doing now. And so we are building the models using training on the healthy subject to make sure that it's unbiased. And we apply that to currently we're applying that to our Wake Forest ADRC cohort and also the external ADNI cohort. What we found is that indeed, and as reflected to some previous studies, the gaps in the dementia subjects are higher and than the cognitive normal, and the gaps in the MCI are actually in the middle. And on top of that, what is interesting we found is that no one has been looking at the negative correlation or the effect of negative correlation when the disease progression changes, because naturally you wouldn't think those are two correlated things. However, what we found is that with the disease progression, for example, the negative correlation in the AD population is actually higher than the cognitive normal subjects. Why it matter? It matters because when we have the negative correlation, the younger subjects are actually predicted to have a accelerated aging, while the older subjects are predicted to have a decelerated aging. That's why actually, if the negative correlation is higher in the AD population, that means that we actually have a reduced detective power, because of a lot of those cases we would consider the elderly population to be younger. However, which is not the case. So we are trying to see whether we can addressing those problems by using the longitudinal trajectories to consider the individual aging trajectories.
Fascinating. Thank you very much.
Thanks so much for the question. I think I see chat. There's some question in the chat.
Yeah, most of these were congratulations. If there's any other questions, please feel free to unmute yourself and ask your question.
I have a question. It's great presentation. Thanks so much for that. So for the multimodal approach you use for using like a PET and MRI imaging data, and you use like a masking approach just to cope with any like a missing data modalities. So I wonder if you evaluate your model, if there is like any like a missing modalities by benchmarking by using like a two data modalities. So let's assume that you have like a two data models for a patient, you use only one versus benchmarking two. And did you see any like a difference in accuracy or any like explainability differences between this comparison? And also was it like a statistically significant, or was it just yeah?
No, great question. Thanks, Abraham. It's actually, yeah, that's what we haven't shown here because, but that's really important because when we are building the models for, so here we are building the model to account for two things. One is better fusion. The other is better cope with the missing data. But the result I'm showing here is only when we have all the data by using those modalities. We also, indeed, we have ablation studies to detect when we are training using these missing modality model and MEMGA models without, I'm testing that without MRI or missing MRI modality and without PET or missing PET modality. And so we are still working on comprehensive evaluations, but based on our current settings, we did see significant drop in some of the modalities. For example, when we're dropping the MRIs, the performance is dropping significantly, but when dropping the PET, it is not. And so I think a comprehensive evaluation would be needed to see whether that's indeed related to compare that to the MRI only models, right? So to see how much gain it is when we are using both only MRI to do the inference, but using additional PET information for the trainings. My hunch is that there will be improvement. However, at the moment, the attention-based fusion mechanisms has limited power to improve the missing modality if the modality is missing for the entire testing population. So yeah.
Hello, Mark Klein here. Can you talk a little bit about how pathology is incorporated into your modeling system? I may have missed it, but is there a point at which you touch base on that sort of gold standard diagnostic tool?
Right. I guess for now, two things. By using PET, we are trying to using it as a surrogate biomarker for any potential pathology happen at the in vivo data. However, for the outcomes for the studies I'm showing here, at the moment we only using the dementia consensus diagnosis as a clinical outcomes, but we are sorting the data where we have a subset of the populations who have the amyloid fluid biomarkers that can be potentially used as a surrogate, as an alternative outcomes of the biological definition of the ADs, and then that can be viewed as a more biological driven or pathological driven predictor models. But that being said, still the fluid biomarkers is again a surrogate, not a like a actual pathology, endophysiology or endopathologies. So in that settings, I think so I know that there are studies from the NAC where they are actually looking at the diseased population, where deceased, I mean people who actually have their brain donations. In that settings, that can be a validation set to see how much people are having amyloid positive versus amyloid negative in these dementia settings when they both have the dementias. I guess because there is a discrepancy between the biological outcomes of the ADs and the clinical outcome of ADs, which the current clinical outcomes hasn't been addressed yet.
Yeah, I'm just thinking about distinguishing between Alzheimer's disease and other dementias and
Right.
Microvascular disease and all sorts of things. But thank you very much. This was a great talk.
Thank you, Mark. Just want to maybe follow up on that is that we actually have a differential diagnosis pipeline, which I don't have time to present here, where we can potentially, if with the pathological driven labels, we can potentially do the classification, or if without the if there are non-AD based dementias, we can using a generative AI approach to model those and to give us a better differentiations for known AD sort of subtypes. So yeah, I think that potentially will be something that we will also be interested in looking into.
Great, thank you.
Thank you.
Wonderful. We have time for one more question, if anyone has one. Okay, perfect. Well, thank you so much, Dr. Ma. We really appreciate this. This was such a wonderful presentation. We hope that everyone enjoys the rest of their evening. This presentation will be posted on our YouTube channel. If you have any questions, please feel free to contact us at care@wakehealth.edu. Thank you, everyone.